I guess we'll get started. Does anyone have any questions about material from last week before we get going on this week's material? Okay, so one quick announcement. So if any of you are in Kelvin's recitation and just want to attend his office hours have moved a couple of doors down. So he's in GHC 7415. The same office as Charlotte for office hours. Just be aware of that. Okay, so I guess we will get going then on the material for this week. So what we're going to be looking at this week is primarily the question of sequence searching and alignment. So how can we take biological sequences and find other sequences that are similar to those, particularly in looking at large sequence databases. Right, so one of the big issues that we need to deal with is that very often when we're looking for sequences, we have a sequence, maybe we've sequenced it ourselves, maybe it's just something we're interested in, and we want to find sequences that are similar to that one. Maybe not exactly the same, but similar, and that can be a tricky problem to solve. It's not so hard to go into a database, take a string and find all the things that are exactly the same as that string. But when you start allowing for some deviation between them, then this becomes a much more challenging problem. So can anyone think of why it might be that we might want to take a biological sequence, let's say a piece of DNA or RNA, and find other sequences similar to that one? What might be some applications of this? Yeah, we might be interested in finding a model organism that has a gene similar to a human gene of interest to us because we can do experiments in a model organism that we can't do in the human. We'll be seeing a lot of other applications over the course of the term, but some examples of these might be that maybe we've sequenced a piece of RNA. So maybe we're doing some environmental sampling, getting RNAs from a microbiome, a gut sample like I mentioned before. We want to know what organism these come from. That might be a case where we're trying to look in a database of DNA sequences and find things similar to an RNA sequence that we have. We might have a newly sequenced genome and we want to know the functions of its genes. This is a general idea known as comparative genomics. We might take the genome we have and try to guess what the genes do by looking for other organisms that have similar and very likely evolutionarily related genes. And then what we would want to be able to do is search our known sequence against similar sequences in databases. It might be that we would use this in various other ways. We could be searching a protein sequence against an RNA database. We could be searching a DNA sequence against a protein database. There are lots of variants on this, but these are just a few examples of what we might be interested in doing that would require us to search for similar sequences. So I'm going to talk first about how we would do this from working with nucleotide sequences, because it tends to be a little simpler in that case. And then I'll get to the issue of how we might modify this to handle some of the harder problems that come up when working with amino acid sequences. But I'll start with the question of nucleotide sequence matching or sequence alignment. And to think about what we are trying to do here to help motivate the problem, what I want you to do is imagine that we've got some piece of sequence that's shown here, AGCT, AGCT. We're interested in going into a database and trying to figure out whether there is anything similar to that sequence in the database. And for the moment, I will imagine that our database just happens to have these three sequences in it. And we'll see how to generalize a bit later. So let's say this is the sequence we're searching with. We'll often refer to that as a query. And these are the sequences in our database that we'll refer to as subject sequences. And what we'd like to be able to do is figure out whether anything in our database is similar to the query sequences which we're searching. So that's going to be our basic task today. Any questions about this yet? All right, so if we want to figure out whether these sequences are similar, a simple way of doing that is to try lining them up and seeing if we see a lot of agreement between the sequences. We can line up our query against the first subject sequence, and we could see that half of the bases are the same between the sequences, and we might think that's a pretty good match, a pretty good what we would call alignment. So lining these sequences up, we get a lot of agreement. We can say that This pair of sequences looks better, we have more agreement. This one looks worse, we have less agreement. So that gives us a kind of qualitative way of thinking about whether our sequences are similar to one another. But unfortunately, it's usually not going to be this easy. You can't just line up your sequences and ask how many bases match. But that's going to be the basic idea behind what we're trying to do. Line up the sequences and look for agreement. To see why it gets more complicated, we can start by considering another case. Let's say we've got the same query, but we now have one more sequence in our database. So HECT, GAGCT. If we try to line up our query sequence against this new database sequence, we can see that we can line it up this way and get 4 bases of agreement. So that's okay, maybe not great. We could also try shifting the red sequence down a bit, get less 4 bases to agree. So that would be an equally good alignment. But we can probably tell just from looking at these that these sequences are much more similar than this alignment is implying. So can anyone tell me why I might claim that these sequences are more similar than this alignment implies? Yeah, so if we just take account of the fact that if we had removed this G, the sequences would line up perfectly, then we can recognize there's actually a lot more agreement. And that introduces the idea of what's called a sequence gap. So we will say that we can allow for the fact that there may be some nucleotides in one sequence that don't align to anything in the other sequence. So if we insert a gap represented by this dash here in our query sequence, we can get 8 bases lining up instead of 4. So that is something we'll usually want to account for, because if we're looking at closely related sequences or more distantly related sequences, it's not so unusual that over the course of evolution you might gain or lose pieces of sequence in one relative to another. So we have to account for the fact that we may have these gaps in these sequences, and that's going to make a lot of the analysis harder. It makes it harder to find the alignments because there are many different combinations of gaps we could insert, and will also make a lot of the mathematical analysis harder. But it's something we have to account for when working with these sequences. So is this clear to everyone? Okay, so moving on, we now have a way of aligning different kinds of sequences. We can line our sequences up, maybe inserting gaps. We can look at how much agreement we get, and we can at least qualitatively say that some sequences seem to have good alignments to our query, some seem to have not so good alignments. But if we want to actually use this in a computational tool and find objectively good alignments or find alignments that are objectively better than others, we need to be more precise. And what we really need to be able to do is to come up with a quantitative way of saying exactly how good this alignment is, or this one, or this one. And to do that, we need to think about an issue that comes up quite a lot in computational biology, and that is having a mathematical model of what we're trying to do. So we need to have some quantitative model that tells us what it means to have an alignment of this type, or this type, or this type. A way of effectively assigning a score to an alignment. And the way we have to do that is by thinking about how good we think it is to have a 2 bases match, or how bad do we think it is to have bases mismatch. How bad is a mismatch relative to a gap? So that would allow us to make judgments like figuring out, is this alignment better or worse than this alignment? Is it better to introduce a gap and get 1 more base of alignment, or is it better to have a mismatch and 1 fewer bases of alignment? So to do this, we commonly use a strategy called an alignment scoring function. And the way an alignment scoring function works, in the simplest version, is that we will have some match score, and it usually doesn't matter what the match score is. We can just say that's +1, we kind of get 1 of these scores for free, and then relative to that, we would have penalties or potentially positive scores for various other things that could be occurring beyond 2 bases matching to one another. So, for example, we might say that if we think matching gives us a score of +1, it's positive evidence in favor of a good match, maybe we'll say that mismatches, so 2 bases that don't agree in our alignment, are negative evidence of value -2. So mismatch is in some sense twice as good as a match. You can put more rigorous theory behind that, so there are ways of talking about what these scores should be in terms of a model of evolution, so how often we think bases might have flipped in the amount of time we're considering to have elapsed between the sequences we're trying to match, but often it comes down to being something more qualitative. That we think mismatches are stronger evidence against a good alignment and matches are in favor of it, so maybe we'll make these a little more negative than these are positive. This is something we need to consider. So the model we're using, the mathematical model, is something we get to decide, and it depends on what we think we are trying to do in matching. So if we are trying to match sequences that we believe are very closely evolutionarily related, so we're taking a new sequence and we're trying to find similar things that are very close to it in evolutionary distance, we would expect to have very few mismatches. So we would say that probably it makes sense for the mismatch penalty to be strong in that case. If we were looking at much longer evolutionary distances, you don't want to penalize as much for a mismatch. Likewise, a gap is something that we would say is evidence against a match. And usually we would say that a gap, so gaining or losing a base, is something that would happen less frequently in evolutionary time than a mismatch. So flipping a base is in some sense less likely to create significant change to the function of an organism than gaining or losing a base. And so probably we would penalize gaps more heavily. But again, it depends on what we're trying to do. So let's say we are taking RNA sequences and we're trying to figure out where those came from in a eukaryotic genome. That might be a case where we want to have a relatively mild gap penalty. Can anyone tell me why that might be? Well, that's a case where we would expect to have significant gaps, because if you're aligning eukaryotic mRNAs to genomes, you expect there to be introns that are spliced out of the RNAs. So you have to have a scoring function that is going to be tolerant of introns, big gaps in your sequences. So that might be a case where you don't want to penalize so much for the gaps. There are other cases where we might want to penalize much more strongly. For example, if we're trying to take a sequence and look at, let's say, other sequences in the same organism that are maybe closely related to one another, in that case we might not want to penalize, or we might want to penalize more heavily for gaps. But to see how this works, let's take one example of the sequence here. So let's say we lined up these two sequences. What we would want to say is, if this is our scoring function, then we would look at the alignments of the bases, and we would see base by base, are we looking at a match? So in the first position, we have an A matched to an A, so we get a match score of +1. In the second, we have a G aligned to a C, so that's a mismatch. We give it a mismatch penalty of -2. We have a match, a match, a match, mismatch, mismatch, match, mismatch. So this is showing the scoring we would put here, and then we could add up the matches and mismatches we got. We could say we've got 4 times +1 plus 4 times -2, which gives us a score of -4. So -4 then would be the number we attach to this particular score. So does that make sense to everyone? All right, so we can take this case as well. We can say that in this case we have 2 matches and 6 mismatches. That would give a score of -10. So we can say that this is objectively worse than this match according to this scoring function. We can see in this case we've got an objectively better score. So 1 mismatch and 7 matches gives us +5. And then the tricky one would be deciding whether this is better than this. And in this case, we could say that we have 8 matches and 1 gap. And for this particular scoring function, we could say 8m plus g is objectively lower than 7m plus x. And so we would say that for this model of how we're going to score sequences, this is a better alignment than this. Any questions at this point? Okay, so again, there is no correct scoring function. We would get to decide our scoring function, and we want to pick a scoring function that makes sense for what we're trying to do with our sequences. So different situations would imply different scoring functions. I will just briefly mention that some of the tools we'll see will use more complicated scoring functions. So, for example, one of the variations you'll sometimes see is instead of just charging base by base for each gap, we can have a more nuanced function that charges one time for starting a gap and then an additional penalty for each extension of that. So in other words, if we have a long gap like this, we would charge a gap open penalty the first time we used to open that gap, and then an extension for each additional base. So this is something that can be useful in cases where we want to penalize heavily for the existence of gaps, but we don't want to penalize too much for how long the gap is. So the example of aligning RNA to DNA would be a case where we might want to do this. We don't expect genes to have very large numbers of introns, but once you've got an intron, it's not so important how big that intron is for deciding whether or not we have a good alignment. Big introns happen, small introns happen, and neither one is really objectively much better or worse evidence for a good alignment. Questions about that? All right, so there are, yeah, so the use of the scores I'll be getting to a bit later. So these scores we can kind of think of as a first pass as a way of attaching a quality to different possible alignments to the same sequence. But really the score is something we'll be using for some more involved statistical analysis. So for the most part, you don't really care what the score is per se. You'll care about the statistical significance of the score, and I'll explain how that works a bit later in the lecture. Does that answer your question? Any other questions? All right, so there are a few other complications, and one of these has to do with what's called a global, semi-global, or local alignment. And to explain the distinction, let's imagine that we are looking at this query against this sequence here. If we line these up as I've described and assign a score to them, what we can observe is we've got this nicely matching piece of sequence here to here, where the red perfectly matches the blue, but then we've got these big gap regions on either end here. So if we go through the scoring as I've described it, we're going to assign a lot of gap penalties here, a lot of gap penalties here, and those will overwhelm the matches. So we'll end up concluding that we have a really bad match between these sequences. But in many cases, we would want to say that this is actually a really good match. It just happens that the red sequence is a subset of the blue sequence. So this might be something that would come up in the situation where we're trying to match, let's say, an exon of sequence against a DNA database it came from. It may be that this exon aligns to one small piece of the genome, and that's what we want to find, that's what we're looking for. But if you penalize for gaps for the entire rest of the genome, then you're going to get a very bad score. So the issue of global and semi-global and local alignments has to do with how we take account of this case, that sometimes we're not looking for matches of an entire sequence to another entire sequence. So what I've shown here is what's known as a global alignment. We insist that all of each sequence matches to something in the other sequence. It may be matching to a gap, but each base in each one sequence matches to something in the other and vice versa. That's called a global alignment. But sometimes we'll want to consider that one sequence may be a subset of the other. And that gives us what's called a semi-global alignment. So the semi-global alignment, we would assume that one of our sequences, let's say the query, is a subset of another sequence. Maybe an inexact subset, but a subset. And so what we do the query, but not penalize for gaps aligned to the database sequence. So in other words, if the query had extra stuff hanging off the end here that didn't match to the database, we give gap penalties, but these overhanging ends in the database sequence we'll ignore. We just won't penalize for those. And that would tell us that this has a reasonable positive score. So you don't see that as much as some of the other alignments, but what you do see quite often is what's called a local alignment. And the local alignment we can illustrate by taking this other example, where this is our query sequence, this is our database sequence, And we might say that we have a good alignment of a piece of the query to a piece of the database, and both of them have extra overhanging sequence in them. So this is actually a very common case. It might come about if maybe we sequenced a new organism, so a new bacterium, and we wanted to know if there were genes in that bacterium that matched any bacterium we already know about. We might take the entire genome of that bacterium, try aligning it against entire genomes of other ones we know about, and we wouldn't expect the entire genome of one to have a match in the other. We'd expect little pieces of each genome to align. And that's where we would want to score based on local alignment like this. So we'd want to be able to figure out if there are subsets of one sequence that align to subsets of another. This is actually the most common case that we'll have in sequence alignment. So usually when we do sequence alignment, we're actually doing local alignment. In some cases, we're doing global alignment, and semi-global you don't see as often, but just worth being aware of what that means when it comes up. So any questions about this? Yes.
In your examples, all your end sequences happen to be the same nucleotide. Is that just an example, or could it be mixed?
Yeah, so my end sequences are repetitive here. It's just an example. It could be anything. The only important distinction is that it doesn't match between the query and the database sequence. Any other questions? Okay, so that basically gives us this essential kinds of alignments, and by this point we should understand what a scoring function and alignment score is. And that brings us to the question that was asked before of what we do with these scores. How do we interpret them? And that comes down to the question of alignment statistics. So the basic issue with alignment statistics is that even when we have a scoring function, it's difficult to determine objectively whether any one alignment is good in some rigorous sense. We can say that relative to this scoring function, this alignment is better than this alignment, but it may be that neither of these alignments is actually good evidence that the query sequence is a significant match to the database sequence. So the query may be completely unrelated to both of these, and we can't tell that from the scores. It may be related to both of these, and again, we can't directly tell that from the scores. So what we really want to be able to ask is this alignment and the score associated with it significant? And we'll ask that as a statistical question. So we'll ask if it's significant in the sense of, is this something that is too good of an alignment to plausibly have occurred by random chance? And if it is, then we'll say it's strong evidence these sequences are actually related. So the way we'll do that is to use some of the ideas we covered last week when I talked about the basics of statistics. First of all, we'll need a statistic, and the alignment score is the statistic we'll use. So those alignment scores I told you to find, we will use as a statistic, so that's the thing we'll quantify our alignment with, and that's the thing we'll use to try to figure out if we're looking at a statistic value that is implausibly large under a null hypothesis. The null hypothesis we'll be using is that we're matching against random nucleotide sequences. There can be some subtleties here with respect to what we mean by random. Usually with nucleotide alignments, what we mean by random is we're just assuming each base occurs with probability 1/4. So if you took your query sequence and just lined it up against a random string of DNA with equal probability of any string, what would the alignment scores look like? And that null hypothesis is going to imply a probability distribution over alignment scores, and we can use that to derive a p-value for this alignment and figure out whether we're looking at a significant alignment. So questions about any of this yet? Okay. So to see how that works, let's take a couple of examples. Let's imagine we've got our query sequence aligned to this database sequence. So we've got an 8-nucleotide query, and we're assuming we happen to find a perfect 8-nucleotide match. Is that actually statistically significant? To ask that, we would take our statistic value. So our statistic is the alignment score, which is 8 times the match score, or plus 8. And we would want to ask, what is the probability we would see a statistic value at least that large by random chance? So under our null hypothesis, we could figure that out by saying the probability we see a statistic value at least as good as +8 is going to be the probability we see a value of exactly +8, because there's no way to get a score better than +8 for this scoring function. And to figure that out, we would go to our null hypothesis and we would ask, what is the probability, just by random chance, that our first space would get a match there? Probability would be 1/4, because there are 4 possible bases there, and we're assuming each occurs with probability 1/4. So there's a 1/4 chance we would get a matching A. Likewise, in the second position, there's a 1/4 chance we would get a G in our random sequence that matches the G in our query. There's 1/4 chance we would get a C in the third position, matching the C in our query, and so forth. So if we're assuming these are generated independently of one another, the probability of getting a score of +8 under our null hypothesis is just 1/4 to the power of the number of nucleotides. So that's 1/4 to the 8, which is 1/65536, or about 1.5 times 10 to the -5. So any questions about this? All right, so things get a little more complicated when we start considering worse scores. So let's take this alignment where we've got a mismatch and 7 matches. In that case, we would want to know the p-value of our alignment for the score s equals +5. And to figure that out, we would say the probability that we're looking for is the probability s is at least +5. And that would be equal to the probability s is +5 plus the probability it's +6. It happens that there's no way to get exactly 6 here, so we can ignore that. Plus the probability it's +7, that also can't happen. Plus the probability it's +8, which we just figured out here, as we know the probability is +8. And we would then want to add those up. If you haven't done a lot of probability, things get a little trickier when you're considering a case like +5, because then we have to consider what are all of the possible ways we could get a score of +5. The only way to get a +5 is to have 1 mismatch and 7 matches, and so the ways this could happen are the 8 ways we could pick the mismatching base, a probability 3/4 of getting that mismatching base. So whatever base we have here that we are saying is the mismatching one, 3 times out of 4, the thing that aligns to it would mismatch, and then probability 1/4 of getting each of the other bases matched. So 1/4 probability to get an A here, and a G here, and a T here, and an A here, and so forth. And then you take the full product, and you get 8 times 3/4 times 1/4 is 7. Question?
Are there errors within this method?
Well, we're assuming we have 8 bases here, so any one of these can be the mismatching base. That's why we need this leading 8 here. So we have to sum over all of the possibilities. Any other questions about this? Okay, so we can add this up. We can get a p-value out of this, and in this case, the p-value is a little worse, but still significant. 3.7 times 10 to the -4th. So we would say that both of these are objectively good matches. They're statistically significant matches. So just one bit of review. Can anyone tell me, are we looking at one-sided or two-sided p-values here? Yes, so these are one-sided p-values. We're only interested in matches that are better than we expect by chance. So alignment scores that are skewed in the positive direction. We're not looking for implausibly low alignment scores. We use a one-sided p-value, and we only look for the probability of the, under the null hypothesis, of the score being at least as large as our specific score. All right, so there are complications here, and one of the big ones is that I've just been talking about alignment statistics for the ungapped case, so ignoring the possibility of gaps. And unfortunately, for the level of detail we assume in this class, pretty much all I can say is that in the gap case, it's much more complicated. So when you start adding gaps, you have to consider the distribution over all of the possible ways of putting in gaps. You're asking for a probability distribution relative to the fact that you put in the gaps in the optimal possible way. The mathematics and the theory behind that gets much more complicated. So we're not going to go through in class here how you would do that. This is actually a research problem. What's generally done for the real tools is to approximate this in various ways, because it's a hard thing to do to really get the p-values exactly right when you allow for this. But just be aware this is an issue, but the standard tools we use that we'll be seeing on Friday will do this for us. They have a lot of this more advanced theory behind this, so you can trust that they're doing this in a reasonably good way. But you really need to get to a more advanced statistics class if you need to know the detail finer than that. But if you understand the ungap case, I think you understand the concepts you really need to use this effectively. Any questions about this? Right, so there's one other really important complication here, and this has to do with what I referred to last week as the multiple hypothesis correction problem. So when we are comparing two sequences, we can get a p-value for a global alignment of those two sequences, and that will tell us if we're looking at two sequences that are similar to a statistically significant degree. But usually we're not comparing just two sequences. We're taking a query sequence and we're comparing that against a database. So we're doing lots of different comparisons, and because of that, a significant p-value for one comparison doesn't necessarily mean that we actually have a significant result overall. So in other words, if we had gotten this alignment of +8 here, which we saw gave us a p-value of 1.5 times 10 to the minus fifth, if we were just comparing those two sequences, we would say that was significant. But if we found this match in looking in a database of 1 million sequences, then we would have to account for the fact that over those million comparisons, some fraction of those, just by random chance, would give an alignment this good. In particular, if the probability was 1.5 times 10 to the minus fifth, that we got an alignment this good by random chance in one comparison, and that's exactly what a p-value means, then we would expect approximately 1 million times 1.5 times 10 to the minus fifth of our million comparisons would be at least that good. So that would mean that the e-value of our comparison would be about 15, or in other words, just by random chance, we would expect about 15 sequences to show up having a perfect match in that database. So this tells us that really we can't call this a significant alignment if we're looking in a database of 1 million sequences. Even a perfect match is not significant there because you expect perfect matches by random chance. So as a general rule, the larger your database is, the stronger the evidence you're going to need, and that's going to make it really impossible to find significant matches to short sequences in big databases. And if you're looking for a problem like this where you need good evidence of similarity, you're going to need some way of getting around that. Either a way of pre-pruning your database, you're making fewer comparisons, or some way of throwing in more evidence, such as lengthening your sequences. This also gets more complicated in real practice. So for local alignments, our number of comparisons has to take account of the fact that we don't just have lots of sequences in our database, we also have lots of ways of picking subsets of our sequences. So again, the math gets a little trickier there. You have to consider all the possible ways of picking those subsequences. But for when we use the local alignment tools, they're going to be building this into their analysis. So just be aware that the e-value is usually what we really need to pay attention to in judging whether an alignment is significant. And the e-value is something we can control with our experimental design, so there may be ways of adjusting your design to give you a better chance of getting a good e-value. Any questions about this? Okay, so that basically is nucleotide alignments. And that brings us to the question of protein or amino acid sequence alignments. The basic idea behind these is pretty similar, so we also have alignment scores, alignment statistics, all of that. But there are some important complications that come up. And to illustrate this, let me take this example, a hypothetical alignment. Let's say we have this query sequence, so this is an amino acid sequence, and we hold up this as a possible alignment, the blue sequence in a database. So if we do our alignment scoring the way I showed you for nucleotide sequences, what we're going to do is conclude that we've got 9 mismatches, no matches. So if we use the same scoring function we had before, we would assign this a value of -18. We would say this is a really bad match. But I'm going to assert to you that this is actually a pretty good match. Can anyone tell me why I might claim that? Yeah.
A lot of the amino acids are similar chemically, like lysine and arginine are across from each other. They're both positive. Glutamic acid, aspartic acid are both negative. They're matched with each other. So chemically, it's probably a very similar peptide.
Yeah, so amino acids are not, well, amino acids can be grouped in various ways based on chemical similarity. And these sequences happen to be lined up in a way where we have a lot of chemically similar amino acids lined up to one another. So S-serine and T-threonine are very chemically similar. They have the same basic groups. Threonine is a little larger, but they could often substitute for one another. You'd expect there would be a high probability that evolutionarily related proteins might put a threonine in one where serine is in the other. Likewise, lysine and arginine are both basic residues. Aspartate and glutamate are both acidic residues. So when we see this kind of similarity, that could actually be evidence that these are closely related protein sequences, even though they don't agree in a single amino acid. So because of this, we need a more nuanced kind of scoring function if we're going to be able to accurately judge similarity in amino acid sequences. And the way we would generally do this is to use what's called a substitution matrix. So the substitution matrix will replace the match and mismatch scores that we saw for nucleotide sequences. And what this is telling us is how similar, or really how substitutable, any one amino acid is for any other in our sequences. So, for example, if we look at a few different amino acids, we might say that some amino acids are more strongly conserved on average over evolution than others. So alanine is chemically similar to a few other amino acids. You might expect that a large fraction of the time, if you have two evolutionarily related proteins and one has alanine, maybe the other one has a glycine, or it has a leucine, or an isoleucine. All of those can often take the place of alanine. So seeing two alanines line up might not be very strong evidence that these two proteins align. On the other hand, cysteine is maybe less substitutable. Cysteine has some important functions only cysteine can do. So many proteins form disulfide bridges. Only cysteines can form those disulfide bridges. So you might expect, if you see two cysteines lining up in two proteins, that's stronger evidence those proteins are related. In some cases, even different amino acids can be evidence of relationships. So D and E, aspartate and glutamate, may be similar enough that seeing an E lined up to a D is actually positive evidence in favor of these being similar proteins, rather than evidence against them being similar. And all of that would be captured in these substitution matrices. So algorithmically, it's not so different. We just plug in the substitution matrix, look up the pair of amino acids, and get a score, rather than just going by matches and mismatches. Now, this does introduce one complication for using these, which is that we have to have a substitution matrix. Usually, this isn't going to be something we're finding ourselves, but we do generally have to pick from a few options that are available. One class of these are what are called point-accepted mutation, or PAM matrices. And the general idea behind how we get these is that you look at many aligned sequences. So you take libraries of sequences we believe are closely related to one another, often at different, what we believe are different evolutionary distances. You line them up to try to find the equivalent amino acids across those sequences. And you ask, how often do you see any pair of amino acids lined up in these groups of aligned sequences? So how often do you, when you look at one sequence in one position, how often do you see an L in one sequence and see an L in another sequence? How often do we see an L in one and a V in another on the line? Encode that in a frequency the f sub LV, let's say, for the frequency with which we see an L aligned to a V over all our pairs of sequences and all our positions of alignment. We would then want to see where that falls relative to the overall frequency of L and the overall frequency of V in our proteins. And if we see that f sub LV is larger than x of L times x of V, that tells us there is a bias towards having L's aligning to V's. This ratio is known as an odds ratio, and that effectively is telling us whether a particular alignment of amino acids is evidence for or against an evolutionary relationship. Usually you'll take the log of this, get what's called a log odds ratio, but basically if the ratio is greater than 1, or equivalently if the log of the ratio is positive, that's evidence in favor of these sequences being related. If the ratio is less than 1, or the log negative, then that's telling you this is evidence against a good alignment. We take the log just so these add up, so you can add up contributions across pairs of sequences, and that's equivalent to multiplying probabilities. So the log odds ratio is the main thing that's going to be used to derive these matrices. And in a PAM matrix, the way this is done is to get all of these log odds ratios and normalize. You get something that's more or less telling you, on average, over a small unit of evolutionary time, how frequently is any one amino acid substituted for any other. We call that a PAM1 matrix. And you can think of that as a way of taking any given sequence and generating a distribution of possible sequences that we would expect that to evolve into a short time later. You can then compose these matrices, these PAM1 matrices, just take products of these to get distributions of sequences longer times later. So if you take the product of 50 PAM1 matrices, or equivalently apply the PAM1 matrix 50 times, you get something that should approximate the evolutionary biases over 50 times that length of time. So with PAM matrices, this gives us one way of getting a model of how often given amino acids substitute for one another, and the larger the PAM number, the longer the evolutionary time we're trying to fit. So depending on how similar we're trying to find our sequence matches, so whether we're looking for closely related sequences or looking for distantly related sequences, we would want, for closely related sequences, a small PAM number, for distantly related sequences, a large PAM number. And the standard tools will generally let you pick from a few different PAM numbers. Another way of doing this, which is pretty similar in practice, is what's called a BLOSSOM matrix. With BLOSSOM, they more or less do the same thing, except that instead of taking all of these alignments and kind of jumbling them together, they will take groups of alignments of more or less different approximate lengths of evolutionary distance. So they'll take a bunch of proteins that are approximately 45% similar to one another, get a matrix from those that they call a BLOSSOM 45, a bunch that are 62% similar approximately, get a BLOSSOM 62, and a bunch that are 80% similar, and get a BLOSSOM 80 matrix, and then you can pick among BLOSSOM matrices to try to balance different evolutionary distances. So if we were trying to take an amino acid sequence and search for very closely related sequences, which of these BLOSSOM matrices would be our best choice? Well, if we're looking for very similar sequences, we probably want a large BLOSSOM number. So if you're looking for short evolutionary distances, you want a high BLOSSOM number or a low PAM number. So again, this is something we would generally pick. If we know that we're looking for distant homologs, we want a big PAM number or a small BLOSSOM number. We know we're looking for close sequences, a small PAM number or a large BLOSSOM number. Often you can use BLOSSOM 62, which is kind of an average one, as your default if you don't really know what you're looking for. So that's kind of a good one to pick if you have no reason to pick anything else. Any questions about that? So there's one last concept I wanted to introduce you to before we break for today, and that's the concept of a multiple alignment. So a multiple alignment is basically what we've just been seeing, except that instead of aligning two sequences, we're going to align multiple sequences. So what we had seen before is often referred to as a pairwise alignment, so one sequence aligned against another at a time. And if we're aligning against a database, we would still be doing many pairwise alignments. One query aligning one at a time against their database sequences. There are some situations in which we want to align multiple sequences simultaneously, and you can think of this as the problem of taking our sequences and inserting gaps potentially in several of them to get a good overall alignment. So to identify the equivalent positions across all of these sequences. We actually just saw one example of where we might want to do this, and that's in the creation of our substitution matrices. So to create the substitution matrices, we need to do these multiple alignments to find the conserved positions across a family of related proteins, and then get from there the log odds ratios for our substitutions. But there are lots of other cases where we might want to do this. Usually this is done by assuming that we are doing a multiple local alignment. It doesn't have to be, but that would be the usual assumption. And there are various ways of scoring this. The simplest ways of getting a score for a multiple alignment are just to generalize the scoring functions for a pairwise alignment. So, for example, we can score a multiple alignment using what's known as sum of pairs scoring. So the idea behind that would be that we would try to find an alignment of all of our sequences that optimizes for the score that you get by summing over all of the pairwise alignments it implies. So if we get this multiple alignment of a red, a green, and a blue and a green sequence, then we would say that to score that, you would take the score of the red sequence aligned to the blue in this multiple alignment. You can figure that out by applying our pairwise alignment scoring function, add in the score of red aligned to green, blue aligned to green, and get an overall multiple alignment score. So we'll see next time that this is actually a pretty important distinction. Algorithmically, it very radically changes how you find the alignment to consider pairwise versus multiple alignment. But it's worth knowing what a multiple alignment is and some of the ideas behind how you might score a multiple alignment. Are there any questions about this?
When you're scoring the multiple alignment, why does when, like in the first excerpt, go split apart? So when like the red and blue are matching, why aren't the C's considered a match?
Red and blue,
and then on the last one, the PGs are matching. The blue and the green.
yeah, actually that's a typo here. So
Okay.
these should be a mismatch here. There's a mistake in the slide there. So the pairwise alignments should be scored as if you're just doing pairwise alignment. All right, but basically that is multiple sequence alignment. Now, before we break, I just want to summarize what you should have learned today is what a sequence alignment is, how we score these, so a scoring function, the distinction between global, semi-global, and local alignment, how we use statistics to assign significance to an alignment, the distinction between the protein alignments and the nucleotide alignments, in particular a substitution matrix, and the concept of multiple alignments. Now I do want to point out that one very important thing I haven't told you today is how you find the alignments. So we've talked about what an alignment would look like, how you would score this and decide its significance. What we'll be covering next time is the question of how we actually find these. So what goes into finding these and what we need to know about that to use these effectively. But basically, by this point, you should at least understand what an alignment is and how we're going to evaluate them. Any questions before we break for today? Okay, so just a reminder, homework is due in lecture on Friday, so please bring your homework, hard copy to lecture on Friday. And I guess aside from that, I'll see you all on Friday.